Welcome to the Mind Institute. My name is John Brown, and tonight I have the pleasure of both presenting the speakers for this evening and also sitting back and enjoying the presentations. The Mind Behind the Minds series is presented as an opportunity for a couple of things to happen. One of them is for the community to learn about what it is that we do here at the Mind Institute. And secondly, for our researchers and faculty to share with others and hear from others about questions they have concerning research and their research program. The third benefit that I have this evening is it gives me an opportunity to go back to school, because since I've been here at the Mind Institute, one of the more interesting and exciting phenomena for me is that every time I come to a lecture, I feel like I'm going back to school, and I just say, wow, that's amazing. Hopefully you will experience that as well, because this is truly an example of what it is we do here at the Mind Institute and with people who are associated with research that goes on here. It is my pleasure this evening to present our speakers. And this evening we have a 2-for-1. Not really. Actually, we have two speakers, but their topics are not unrelated at all. We have presenting for Dr. Judy Vanderwater, Daniel Braunschweig, who is a researcher in her lab, and he will be talking about the immunology of autism. And as you know, there are many, many questions and concerns about immunology and autism, and he will be sharing some of the findings from research in Dr. Vanderwater's lab about this particular topic. Our second speaker will be Dr. Isaac Pessah, who is a long-standing faculty member here at UC Davis and at the Mind Institute. He is the chair of the Department of Molecular Biosciences and the director of the Center for Children's Environmental Health. And he has a long history of research regarding environmental toxins and other agents. And tonight he will be particularly talking about that research as it contributes to the development of models that might help us understand more about this disorder we refer to as autism. So I give you Dan Braunschweig. Thank you.
Well, good evening, and welcome to the Mind Behind the Mind. This evening, as Dr. Brown mentioned, I'll be discussing immunologic abnormalities in autism. And there's going to be a lot of data that I'm going to present, so try not to get bogged down in it. The point of this is that autism is a heterogeneous disorder. There's a whole range of autisms. And by looking at the different immune manifestations, we're hoping to distinguish between different ones. So first, a bit of introduction on autism spectrum disorders. As I mentioned, they're highly heterogeneous, complex variability of symptoms and defined exclusively by behavior. So the core defining features of autism, the three main features that are used to define, are impairments in socialization, verbal and nonverbal communication, and repetitive stereotypical behaviors. Okay, so here's the last introduction slide about autism, but here are some points. Some of them are fairly recent. I'm sure many of you are aware of the increase in incidence of autism. One of the latest estimates showed 1 in 150, and interestingly, the male to female ratio has held up over quite a long period of time. So it is known that autism has a strong genetic component. So don't feel that the immunology of autism is a distinct aspect of it, but the immune system is completely controlled by genetics, and so these two things are totally integrated. Okay, so this led us to look at autisms in terms of the causes, the upper upside down triangle there, genetics, perinatal exposures, some other environmental triggers, perhaps some susceptibility, and all of these things, or some of these things, may combine to lead to an autism spectrum disorder, which then has these behavioral outcomes, as you'll see below. And so the behavioral outcomes that define autism are not the only effects that you see among patients with autism. There are certainly other things that go along with it, including seizure disorders, and as you'll see in this talk, immune dysfunction. Okay, and this constellation of behavioral phenotypes is how we like to think about autism spectrum disorders. Okay, and now for the immune system. So you all know that to be healthy, you need a functional immune system, and it's also important to recognize that the immune system plays a fundamental role in neurodevelopment. There are interesting examples from animal models where in mice with a highly compromised immune system, you have substantially decreased capacity to learn behaviors which are normal in immunotypical mice. And then furthermore, if you take a normal mouse and you deplete its adaptive immune system as an adult, it will show a significantly decreased ability to learn new behaviors. And so you can see that both for neurodevelopment as well as for maintenance of proper neuronal function, the immune system is fundamentally important. And throughout life, there's a lot of crosstalk. There are molecules which have fundamental roles in both the nervous system and the immune system, and cells as well. So there's a lot of interplay. Okay, so now here's a quick rundown of what the literature has to say about the connection between autism and the immune system. And essentially what you'll see from this slide is that there is a lot of conflict in literature about the degree and extent and direction of immune dysfunction in autism. There are some studies that show enhanced autoimmunity, so in other words, a hypersensitive immune system, others that show reduced immune function. So the difficulty, of course, in defining a study group when doing a study in autism is the fact that this is a disorder that is characterized exclusively by behavior. There isn't yet a blood test or a genetic test that can be done to very specifically categorize individuals with autism. And thus it's difficult to develop a cohort of individuals that you would compare in a study when looking at these biological markers in the immune system. And so, and then furthermore, the last point there, which is important as well, is that immune dysregulation or dysfunction is certainly not observed in all cases of autism. Okay, so here's the work that we are currently undertaking in our lab, looking at the immune system in autism. So first I'll go through the immune system in children and patients with autism. And we're looking at immunoglobulin levels, those are antibodies. And then, so not only the levels of antibodies, but also the specificity or what the antibodies bind to in terms of autoantibodies and binding to self proteins. And then afterwards, I'll show you a few slides on maternal antibodies in autism. Okay, so here's a recent publication from our lab where we discovered that among some children with autism, we saw decreased levels of immunoglobulins. These were immunoglobulins G and M. And IgM, immunoglobulin M, is the first antibody or immunoglobulin that's produced in an immune response. And IgG is the most abundant antibody in circulation. So antibodies are molecules which your body develops in response to a pathogen or in response to the signal that a pathogen is attacking. And they're fundamentally necessary in an adaptive immune response for clearing that pathogen and for getting better after you get sick. So IgG and IgM are the two main classes of circulating antibodies. And we see significantly decreased levels of IgG and IgM among a large cohort of children with autism. So this was part of the CHARGE study, where we recruited a large number of families with children with autism that were all age matched, and which is an important thing for me to say, because the thing is that as children are growing, their immune system is developing very significantly. And so it's very important that if you're going to do comparisons between groups of children, that they be very precisely age matched. So that was a very important part of the study. So those differences were significant, but even more interesting was when we looked at the total IgG levels compared with the ABC score. So the ABC is a test, the aberrant behavior checklist, and it's the higher the score, the more severely affected a child's behavior is. And so interestingly, we saw a negative correlation between IgG level and score on the ABC. In other words, the lower the level of circulating IgG in the child, the worse their behavior was. Okay. Now, looking at those same antibodies, but this time in terms of what their specificity is, what they bind to, we found that among children with ASD, there's a subset of them who have autoantibodies, antibodies which could or would bind to their own brain tissue. And that is an interesting finding, and there have been several studies reporting this. We undertook both immunohistochemical studies where we would look at slides, tissue sections of, in this case, rhesus brain, and also by Western blot, looking for the binding of these antibodies to both the tissue in terms of what cells they bound to, as well as generally just looking for what proteins the antibodies were binding to. So if you take a look at this slide, A, B, and C are of the same tissue but with increasing magnification. This is in the cerebellum, and those labeled cells are stained with the antibodies from children with autism. And interestingly, when we stained this monkey cerebellum tissue, we saw a very specific staining of a particular cell type in a particular area in the cerebellum. So these are Golgi cells, they're interneurons in the cerebellum. We're not sure what the role of these antibodies is, or whether they have a pathogenic role or not, but it was very interesting that we noticed staining of Golgi cells in about 1/5 of patients with ASD and not in any of our typically developing controls. And so that we're still working to try to figure out what this means, but it was a very interesting observation. We were very surprised to see this staining. This would be the staining that you would expect if you had a highly specific antibody for a particular cell type, and so it was kind of shocking to see this. So then another way of looking at antibody binding is by the Western blot, where you separate proteins by molecular weight, and then you probe them with antibodies. So the band that we're interested in here is that 52 kilodalton band, and among all of those children who produced that staining of the cells that you saw on the previous slide, they also had this band at 52 kilodaltons, which is one more piece of information that we would need to try to determine what the protein that those antibodies is in fact binding to. But so you can see that the left two strips there were probed with antibodies from a child with autism, and then the TD and DD are controls, a typically developing control, and a control with non-ASD developmental delay. Okay. So in addition to seeing these autoantibodies against cerebellum, we also saw them against thalamus and hypothalamus, two other brain regions. And interestingly, the staining pattern was a little bit different. The strip marked A1, furthest to the left there, is the typical staining pattern, where we see a series of bands at about the same molecular weight, but also some smaller ones. And again, these were significantly associated with a diagnosis of ASD. Okay, so those were the antibodies, and now we're going to look at the cellular immune response. We're involved with several collaborations at the Mind Institute here, and so there's a cytokine called leptin, which has received some amount of press recently, and we noted, interestingly, that when you looked at all ASD cases, we didn't see a very significant difference between the leptin levels, but when we separated the early onset and regressive phenotype, we saw significant differences. And so one of the points of what we're doing here is an attempt to subclassify autism, and this was a very interesting finding here, where we found that among the early onset phenotype, that we saw increased levels of leptin in the plasma. So this is definitely a goal of this research, is to be able to come up with tests that we can give to at least attempt to tailor therapies for these children to the form of autism that they have. This complicated slide here shows several more of the cytokines that we looked at and the differences between the regressive and early onset phenotype. So ultimately, it's likely that there are some differences in the causes of those different phenotypic outcomes, and it is interesting to note that when looking at cytokines, which are the chemical mediators of the immune system and the regulators of immune function, that we see significant differences between children with those two phenotypes, and then certainly differences between them and typically developing children. And so this is, these are data from charge samples, including those currently enrolled in the Autism Phenome Project. Okay, and so there's another regulatory cytokine that we looked at, transforming growth factor beta, and this also we compared with behavioral outcome among the children. And so you can see here that TGF-beta levels were significantly decreased in children with autism compared with the typically developing controls. So interestingly, we also enrolled siblings of children with autism in a lot of these studies. And as a side note, both in this study as well as several others that we've looked at, when we include the siblings, they often have an intermediate phenotype in terms of levels of things that we're looking at, which is just another interesting note here. As you can see on the right with the siblings, this is a clear example of it, being intermediate between the autism and general population. But this phenomena occurs quite often in these studies, thus suggesting the genetic nature of this disorder. Okay, so, and we saw a negative correlation between behavior and levels of TGF-beta. So this is sort of the second time that we've seen a trend that is related to behavioral outcome. This is a slightly different behavioral test than what we showed before, but a similar outcome. Okay, and so just to continue with this TGF-beta, which we've just been looking at and saw the correlation with behavior, TGF-beta is an interesting cytokine which has many and fundamental roles in neurodevelopment and the nervous system, as you can see here, and then also in the immune system. Everything from wound healing and immunosuppression to neuronal survival. Okay, so now moving away from the children a bit, we're going to be looking at the immune system of the mother, and first the role of maternal antibodies to fetal brain in autism. Now, it's important to note that during pregnancy, the developing fetus doesn't have it does not have a developed immune system. And the antibodies that are circulating in the mother's circulation do enter fetal circulation during gestation. And in fact, at birth, all of the IgG that is in circulation of the newborn is maternally derived. And furthermore, there is a higher level of IgG in circulation in the newborn than in the mother. So there's an active transport of IgG across the placenta. Remember that immunoglobulin G is the most highly abundant immunoglobulin in circulation. So the possibility exists that autoantibodies, IgG, which is directed against self, could have a deleterious effect on the developing fetus. And there is precedent for that in other autoimmune disorders, such as lupus. So we decided to look at whether mothers of children with autism had autoantibodies against fetal brain proteins. And lo and behold, we found that among mothers of children with autism, we saw this characteristic pair of bands that was present in about 12% of the initial study, and we've actually replicated this a couple of times now. So a small but significant fraction of mothers of children with autism have autoantibodies against fetal brain protein, and specifically against proteins at a molecular weight of 37 and 73 kilodaltons. That's really just mostly relevant for trying to determine what these proteins might be. So you can see in the first three strips on the left examples of the leftmost one has only the 73 kilodalton band, and then the next two, the next one has 37 only, and the third one there on the right has both. So we saw this in about 12% of mothers of kids with autism, but when we looked at the behavioral phenotype of their children, we saw that of the 7 who had the pair of bands, 6 of them had the regressive phenotype, which was rather surprising. And among typically developing controls, as well as controls with non-ASD developmental delay, we didn't see this pair of bands at all. So this is a summary of the paper. In fact, both the 37 and the 73 kilodalton bands alone were also significantly associated with a diagnosis of autism. And so in total, about 20% of mothers of children with autism had antibodies against one of those two molecular weight proteins. So this, that work that I just showed was also replicated in a prospective study where blood was taken from a large cohort of mothers in Southern California during gestation. And then after their children were born, those with ASD were identified. And from that cohort, from the larger cohort of, this was all women who delivered children in Orange County over a 2 or 3 year period. Of all of the ones who later developed ASD, age-matched controls with typical development were selected from that larger pool so that we could compare markers in the mother's blood during pregnancy with controls, but blood that was taken during pregnancy before the diagnosis was known. And again, we had essentially the same results or very similar outcomes with regards to the banding pattern and its association with an outcome of autism. In this case, we also saw a 39 kilodalton band. It's a little bit difficult to finally resolve the molecular weight of proteins by Western blot, but it was very interesting to see this finding replicated in a totally independent cohort. Okay, so here's a summary of the maternal antibodies to fetal brain. We found a specific subset of mothers have these antibodies, and that they are highly associated with regression. We have seen this in 2 studies in our lab, and then also it has been replicated by other researchers in other parts of the country. So this is a very interesting finding, and ultimately may have some role. It's important to note here, though, that what we're seeing here, and I don't mean to say that any of this has a causative role. We don't know this yet, but ultimately it is an interesting finding, and we're working actively to determine whether this is a causative role or just an association with autism. So in order to answer that question, we have some animal models going on right now, both primate and mouse models, to see whether we can induce behavioral outcomes in the offspring of animals administered IgG during pregnancy. And so we started this with a pilot monkey study where we had several animals who were exposed to human maternal IgG during pregnancy, and then the behavioral outcome in their offspring were monitored. Now, this was a small pilot study. We had, of the two groups which were exposed to IgG from mothers of children with autism, in group A there, we only had 4 live births. Now that's, the group A was the 37 and 73 kilodalton band. It was those mothers, which I showed you previously. And that's the group that I'll show you the data from. We only had 2 live births in the other group, and it was not, so ultimately we are currently in the process of repeating this study. But ultimately we tested these, the young newborn monkey, or the young monkeys for several criteria, which you can see there on the bottom. And interestingly, we found very significant outcomes. And and I'm going to show you a quick video, but take a look at this. This is the number of midline crossings that is in the mother preference task, which is, so the young monkeys are reared by their mothers until they are weaned, and so they recognize their mothers, and they'll tend to go to their mothers. And so the mother preference task puts 2 mothers on either side of a large open cage. One is the mother of the monkey that's being tested, and the other is an unknown mother. And the task is just asking whether the monkey recognizes their mother and goes to her. And so let me show you what happened. Okay, so this is a typical monkey. It started running a little bit, found its mother, and is just going to spend time with her. And this is what we would expect. Okay, and so this was the offspring of a monkey who was injected with the autism antibodies. And again, his mother is on the left. And okay, so ultimately, that was an example of the mother preference task. There were several other tasks that also showed significant differences. This was very intriguing pilot data, and so we are currently replicating the study with larger numbers to see if we continue to observe this. So another one of the tests that we looked at was the social dyad. This was after the monkeys were weaned. They have groups which they are housed with after they're weaned, and so this test was just to look at how the monkeys behaved with another monkey of the same age that is familiar. And ultimately, the stereotypies that we were observing included backflipping and running around the cage. Ultimately, during the observation, normal monkeys will hang out with each other, and they'll run around, but they'll do things together, and they'll interact with each other. And essentially, the summary of that larger bar that you see on the right there with the autism IgG is that the young monkey, the offspring of the mother who was injected, didn't do anything. It didn't spend time with its familiar monkey in the cage, instead just spent a lot of time doing backflips and running around on its own. So this is not to say at all that we made autistic monkeys, but it was interesting to note that we saw some significant behavioral differences after this treatment. Okay, so of course the fundamental gap in this maternal antibody story is determining what the antigen is, what the protein is in the fetal brain that the mother's antibodies are binding to. We know that it's fairly specific for fetal brain. We've looked at many other tissues, but we're still working on trying to figure out what the identity of the protein is. And then after we do that, that will hopefully inform our studies about how these antibodies may, what their role may be in the development of autism, or whether it's just a side phenomena, which also may help to elucidate a potential mechanism of developing autism. And then, of course, the ultimate goal would be to devise a therapy based on this. And that is what I have for you. Here are all of our lovely lab members, and thank you very much.
Good evening. I apologize for being a little late. I got held up on the causeway, so fortunately it wasn't any bad accidents or anything, just somebody with a flat tire. So as I'm warming up the computer here, how many of you become more aware of what you're being exposed to in the environment? Just over the last, let's say, 5 years. You don't have to think that far back. So what do you think the primary reason for your awareness is? I mean, just shout it out. Do you think media? Media? You have much more information that you know what to do with, I guess, and every new finding gives you cause for concern. But I think if you think about it, we have about 80,000 chemicals that we know are in production. They're essentially anthropocentric. They're not necessarily found in nature, although close relatives could be found in nature. Some of them we think are innocuous. They certainly come up negative in many of the toxicity tests that we perform. And so we generally think they're safe. At least we have certain criteria by which we deem them as safe. The question is, what is the dose at which we're exposed? What is the timing at which we're exposed? And what are the combination of chemicals that may individually not cause harm, but in combination cause a problem? And so what I'd like to talk about is some of the methods that are used to assess potential risk to human populations, and also it goes for animal populations as well. What are some of the shortcomings? Have you all heard of dioxin? To some, it may strike a chord because it made the news not too long ago, long after Vietnam. Somebody tried to poison the Ukrainian president by putting a little bit of dioxin. And the key word here was a little bit. And they almost succeeded. In fact, he has developed some very debilitating chloracne in his face. It was excruciatingly painful for quite a period of time after the exposure was performed. And the poor fellow has about 1,000-fold higher chance of developing liver cancer. So this is with one exposure on one attempt to do him in. Now, we regulate compounds like dioxin very heavily. In fact, we have tests for them. I'll get to the slides in a second. So we regulate them very heavily. We have tests for them. We now screen for compounds that have dioxin-like activity. And if in fact you find that they have very potent activity, what happens is they find it much harder to reach the market. Do we do this for compounds that don't look like dioxin or don't have any activity like dioxin? You'd think we should, right? But what do we test for? You have to test for something. That something has to be relevant to human risk. So here's kind of the problem in perspective. If you don't have tests for something and you generally assume it's safe, you may let some very, very harmful chemicals through to human exposure. You can all think of some examples. Anybody have an example? Thalidomide. There we actually did test thalidomide in animal studies. The problem is we used the wrong species and we were looking at the wrong endpoint. So as soon as we realized what had happened, of course, it was withdrawn from the market. That's a very, very pronounced effect. It's a teratogen. You know exactly what happens, and you can relate it to exposure. But there's now general agreement that about 3% of all developmental defects are attributable to exposures to toxic chemicals. That's a large number of individuals. 25% of these defects are thought to be due to gene-environment interactions. That is where individuals, because of their genetic diversity, have susceptibilities that may not be found as a general trait throughout the population. We think that this is an underestimate, and the reason we think this is because we only score neurological defects if they're severe enough or close to birth. If somebody develops schizophrenia at age 14 or 18, we don't think of it as a hit in utero or a hit during the neonatal period. We think of it as something that develops and we quite don't understand it. We have some clues. But that it couldn't possibly be something that occurred during development. So the two are usually separated, and that's probably not the case. So Dan probably introduced you to autism, and to tackle autism in terms of how could autistic individuals or individuals that are at risk for autism be susceptible to environmental insults is a very complicated issue. We know that autism has very high heritability. We know that multiple genes confer risk, and this is becoming more and more difficult because association studies don't always replicate. So one group finds one association with a particular marker or a gene, and others don't replicate it. And in fact, now we think, well, you know, if you put all the literature together in what we call genome-wide screens, that there are probably many, many genes that contribute to autism risk. And it's probably not the same genes or cadre of genes in every individual. We think that there are epigenetic mechanisms that can contribute to susceptibility. This is where the DNA methylation is different in every individual, and some patterns of methylation may be more at risk than others. We also now have this intriguing, very recent finding within the last 5 years that in fact, even though there seems to be heritability, the association with the genome is not that of the parent. So you can't find the same mutations or polymorphic characteristics in the parents, but they're somehow de novo. So they're created in the offspring, and you can measure them and you can quantify them, but they're not necessarily transmitted through the parents. And we don't know how this happens. Sometimes we think that this just arises spontaneously. Some areas of the chromosomes are more susceptible to others. So given that autism has a very complex heritable and perhaps environmental basis, could we perhaps do a general global hypothesis, propose a general global hypothesis that individuals that are at risk for autism are particularly susceptible to environmental risk because they have so many, let's say, unique genes that contribute to an adverse response or an inappropriate response. And so that's what this says. I just said it in words. So this is on the chemical side. There are 1,000 commercially important chemicals. How many of these do you think we have adequate data for? I was at a meeting last summer where somebody from the National Toxicology Program told me how many and what the cost has been. And it was really quite sobering because I didn't expect it to be. So for about 11 compounds, we have very good risk assessment tools. We have many others that are sort of incomplete, but we have some idea. But those 11 or 12 compounds cost us $3 billion to get there. That's amazing. I mean, that's an amazing cost. That includes studies like in non-human primates, studies in rodents, studies in tissue culture. So if you think about 80,000 chemicals and having to get, really good data on 80,000 chemicals, the stimulus package isn't going to be enough. So the vast majority of commercially important chemicals actually lack adequate toxicity testing. We can screen them for mutagenicity, and that automatically takes them out of the picture, at least it should take them out of the picture. We test for teratogenicity, we don't test for very subtle neurodevelopmental outcomes. We've just started, especially with pesticides. So the EPA has identified priority pollutants that they think have to have data, and they should be put up above other chemicals. So as you can see here in drinking water, they've identified about 7,300 chemicals that they need to get more data on: antimicrobials, pesticides, high volume chemicals, HPVs. Inert pesticide ingredients, we always perseverate on the active ingredients in pesticides because they're typically neurotoxic, but we have lots of things that go into the formulation that we have no idea if they're actually also neurotoxic or have other effects. Add to this the fact that you are never exposed to a single chemical, that you're exposed to mixtures, so we really need to address the issue of combinations of chemicals, which becomes much harder for the scientist. Do chemicals alone that don't seem to have an effect when put together have an effect? So that's synergism. Or can one chemical negate the effects of another? Antagonism. We also don't pay much attention to relative timing. So if you expose an animal for, let's say, trying to predict neurodevelopmental toxicity from about you expose the mom 2 weeks before conception, and you keep the exposure going throughout lactation, and you say, I don't see anything. But then you change the protocol, and you only expose them during lactation, and you see something. Timing is important. So the way that we try to muddle through this, and this is, again, what we're faced with as toxicologists, as epidemiologists, is this idea of risk assessment. So in order to identify a relationship between exposure and outcome, you use epidemiology, and use epidemiology on human populations. That only gives you an association, though. So DDT and the risk of breast cancer, and you put an odds ratio to that. So you're 5 times more likely to see breast cancer if you're pesticide levels, DDT in this example, are at a certain level or above. More recently, what we've tried to begin to do, and there's a real effort at here, is to, so these studies are very expensive. So let me ask you this. So is there a correlation between DDT and autism? Has an epidemiology study shown this? Has an epidemiology study shown that PCBs confer risk for autism? Well, if you calculate that autism occurs 1 in 150, which is quite much higher than we originally thought, what sort of study group, how large a population would you have to study? The cost of such a study would be quite high. So with the National Children's Study, we're trying to get a large population-based study of 100,000 individuals from essentially birth through young adulthood, but it's 100,000 kids. So you can calculate how many of those just by the current rates would develop autism. So that's probably not a large enough study, even though it will cost us 1.1 billion or so. Not that it's not worth it, because it's going to tell us a lot about heart disease and cancer and environment, gene-environment interaction. So people have turned to in vitro and in vivo animal models. In vitro now is really turning the corner because we usually use in vitro in cell culture. These cells usually originate from animals. Animals aren't always predictive of the human condition. So what has happened in the last 2 years is that we've identified a way of taking somatic cells from individuals and pushing them back in developmental time to stem cells. Because you know a lot about the individuals from which these stem cells, these original cells originate, you assume, and it's been actually recently validated, that the problems that go along with the genetics go along back in time with the stem cells. And then when you ask the stem cells to become something that you're interested in, like a neural cell, those problems should come along with it. So for a complex disorder like autism, where you have many, many genes, this actually may be a really, really intensely informative way to proceed. And this is something that is now just being developed. But as I'll show you some data for animal models and in vitro, in particular some compounds that we've been interested in. We don't have any data to show you with human stem cells at this point. So the overarching hypotheses that we use is that genetic susceptibilities play off of environmental exposures, and these environmental exposures, if timed correctly, will influence autism risk, the severity of autism, and the treatment outcomes. So how can do we have any evidence? So if you think about this sort of conceptual model here, here's genetic susceptibility, and there could be many, many genes here that are either what we call defective or unique. Environmental factors play off of these genes to enhance or actually increase impairments or enhance the autism phenotype. And some of these environmental factors actually could impair natural repair mechanisms, which would then lead to a more dramatic impairment. So let's talk about the nervous system. You heard about the immune system and possible risk factors. So one of the common features of several neurodevelopmental disorders, including autism and autism-related disorders, is this idea of the developing nervous system lacks a balance. And so what do we mean by that? We mean that the nervous system is essentially controlled by excitation and inhibition. And the organization and the timing of excitation and inhibition have to be precise. Otherwise, if excitation dominates, you can develop something as overt as seizure disorder. If inhibition predominates, you can develop some form of narcolepsy where you just fall asleep, like I'm putting many of you to sleep now. So what we think about is this is a very general concept, and this review by Belmont and Bergeron really bring this home. It basically says, look, we know of many genes that have been associated with autism and autism-related disorders, and these are the kind of patterns that we would expect in any of these disorders, where in some cases the connections between the neurons, so the number of lines here between the balls, are deficient or they're overconnected. That would influence the balance of excitation or inhibition. Or the cells that are inhibitory could die off prematurely or be underrepresented, and so you have more green than red, and so in fact you're overexcited. There are too many excitatory neurons over inhibitory neurons. Or the opposite. Or you can change the way in which the density progresses, so you don't have pruning of neurons and loss of neurons when you should. Or you may have some other overarching neurochemical like serotonin, which changes just the overall reactivity of a normal number of neurons. So, how does this relate to environmental exposure? Well, it turns out that most of the pesticides, so let's just take one group that's heavily regulated, one group of chemicals, pesticides. Most of the pesticides actually act by upsetting the balance of excitation to inhibition, and they do so in very, very clever ways. One is to disrupt the cholinergic signaling system. So acetylcholine is one of the most common neurotransmitters in your peripheral nervous system as well as your central nervous system. So everywhere you see one of these ACHs here, that's the neurotransmitter that communicates the presynaptic side to the postsynaptic side. So acetylcholine is a very common neurotransmitter in many parts of the nervous system. There's an enzyme which exists within these synapses called acetylcholinesterase. So as I'll show you, many of the insecticides that have been developed through the years, and some of the most heavily used, are acetylcholinesterase inhibitors. The way of which they kill insects is to inhibit acetylcholinesterase. The other key player here is the target of acetylcholine, which is a receptor on the postsynaptic membrane, which is called the acetylcholine receptor. Typically, the types that we'll talk about are actually excitatory receptors, so they enhance excitation. Organophosphate insecticides, such as chlorpyrifos, in fact, are acetylcholinesterase inhibitors. So chlorpyrifos was banned about 7 or 8 years ago from home use because a group in Colombia showed that it had negative impacts on birth weights in homes that heavily used these for cockroach control. And so this was a major factor in getting this ban. It's still heavily used in agriculture. And in the United States, there's about 1 billion pounds of organophosphate, not just chlorpyrifos. There's about 200 of them that have been registered for one use or another. But they all fall in the class of organophosphate, about 1 billion pounds that have been applied yearly. Diazinon is another. So these two inhibit acetylcholinesterase, which increases the activity of cholinergic synapses, many of which are excitatory synapses. So you can think of there being an increase in excitation relative to inhibition in exposed systems. Now, there's a 1-2 punch. Remember I said mixtures. There's another group of compounds which has just come on the market within the last 20 years. So organophosphate have been around since about the 50s and 60s. They really came on board in the 50s and in the 60s. They're still with us, of course. But more recently, in the last 15 years, there have been what are called neonicotinoid insecticides used. Now, instead of inhibiting the enzyme, these guys bind to the receptor that I told you about and enhance its activity. So here you've got a group of compounds that inhibit the enzyme that should break down the neurotransmitter, therefore causing hyperexcitability. And here you have compounds that were modeled on nicotine, which enhance the receptor's activity. So are these safe? Well, for organophosphates, we know that they're extremely toxic. So we can control insects with them, but you've got so many warning labels, as you probably are aware, that if they're organophosphates, they say you have to be extremely careful. Okay? And we are concerned that overt neurotoxic effects have been essentially reported in humans from using these pesticides. But what about these? Well, you can be afraid of them or you can understand how they work. And so I was afraid of them initially, and then as information came out, it became clear that neonicotinoids, and these are molecular models based on receptor interactions, that the major neonicotinoid insecticides are really great at targeting the insect receptor, but don't really target the human receptor very well. And so the idea is you can use them to target insects, so you put them on your dog, the fleas fall off, but they're much less likely to create overt toxicological problems, even if you're exposed to them. Why? Because the human receptor really isn't a good target for them, okay? So there's a margin of safety there. The problem with that concept is that all of the data that was shown to be selective for insects over human type or vertebrate receptors was all done with one type of receptor that was cloned into a heterologous system. In other words, into a non-expressing cell, a cell that doesn't normally express the receptor. And it turns out that this is the major form from the chick, small chickens. So it's present in the mammalian nervous system, but not in very high levels. What we don't know is if all of these different forms of the receptors here, from alpha 2 to alpha 10, beta 2 to beta 4, whether these represent better targets for the insecticide. That is where our gap is. We don't know whether all of the receptors that the human brain can express are not targets for these insecticides. That remains to be seen. Okay? Why is this important to autism? Well, we know that in autism there are reports, and they've been replicated at least in a few labs, that acetylcholine receptors, those receptors that can recognize the nicotinoids, are altered, that they are actually less expressed in autistic brains than in comparison brains that don't have autism. And so the question arises, could this be one way in which children with autism might be more sensitive to these compounds? Again, we don't know that, but it certainly begs a question that needs to be studied. Is there epidemiological evidence that organophosphate may be associated with developmental problems? Well, in animals, we know that there's lots of evidence. In humans, we know that there is evidence that they may cause cognitive problems. But in this study, just down the road at Berkeley, Brenda Eskenazi and her group essentially have a very well-studied population of farm workers in Salinas that are highly exposed to organophosphates. And they asked a very simple question. Is the level of organophosphate in the urine and in a later analysis in the blood associated with a higher incidence of, in this case, they didn't score autism because it's not a large enough population, but with pervasive developmental disorders. This came off of a checklist, so it has its limitations, and I want to caution, and she cautions, that you can't overinterpret these results. But in fact, there seems to be an increased risk for having a child with pervasive developmental disorder, or signs of pervasive developmental disorders, as the levels of organophosphate increase in the urine in this case. So this suggests that these can have more subtle neurodevelopmental effects than had previously been thought. What could be the mechanism? Well, in a study from a group in Italy back in 2005, they essentially reported that a particular gene had a polymorphism, and it turned out to be the PON1 gene, which is a peroxygenase 1 gene. The protein codes for a peroxynase, which actually metabolizes organophosphates. And they went on to suggest that the activity of the polymorphic version of the protein is in fact less active or underexpressed, suggesting that people with these polymorphisms would have a decreased ability to metabolize the active product, the organophosphate. The study has been replicated in 2006 by a group in, it's not Hungary, it may be Hungary, and again in 2009. So here is a possible scenario where you could have gene-environment interactions, not that it's causative for autism, but could define a higher susceptibility if you are exposed. And that is the altered expression of nicotinicholinergic receptors, let's say fewer of them, which may represent a genetic susceptibility. Nicotinoids and organophosphates would affect this system maybe more severely with people with these alterations. And in folks that have both the decreased receptors and the decreased enzyme that degrades the chemical, you may work in an additive or synergistic way to enhance the potential negative outcomes. Okay, so that's just one way to look at it. A lot of this has to be supported by data. The data that we have is very preliminary. So I just want to finish off, because I'm running out of time, about some polychlorinated biphenyls. You've all heard about polychlorinated biphenyls. We banned them back in the mid-'70s, but they persist. And those that persist in the environment turn out to be not the ones that we originally worried about, the dioxin-like PCBs. They're the so-called non-dioxin-like PCBs. So about 15 years ago, when I gave a talk about our new findings at the EPA, they were extremely pleased, but in private, they came up and say, "Why are you studying these PCBs? They're like, who cares?" Right? They're not dioxin-like. So I want to share with you some of our recent information about non-dioxin-like PCBs, and then I'm going to segue. I'm going to skip some slides because I don't want to overburden you with data. But I really think that the idea is that we're going now from a new target, which really which recognizes non-dioxin-like PCBs, and we've gone from the target all the way out to what the neurodevelopmental consequences could be, and we've filled in between. The target that we're looking at is a common currency in cell signaling. It turns out to be whether or not these compounds alter calcium signaling inside cells. So in this neuron here, you can listen in with special techniques on the little signals that go on in terms of calcium going up and down. These are meaningful messages to the cell to grow or to let out a synapse or to let out a dendrite, to let out a spine. And these you can actually measure, and in normally developing cells, they're very precise and they're very local. They regulate everything from signal transduction to the metabolic rates of the cell, to gene transcription, to the growth of the cell, as I just mentioned, to the movement of cells, and also to their programmed cell death, which is important in pruning cells in the nervous system. Why is this important to autism? Well, it turns out that this is a short list of genes that have been associated with, single genes now, associated with autism. Some of them are very rare, one of which is this one up here, which is a calcium channel itself, which has just a mutation at one amino acid. Individuals that have this mutation have a syndrome called Timothy syndrome. They have heart problems, but they also have a 60% autism rate. Okay? And these are some of the other genes that either regulate calcium or are themselves regulated by calcium. So it may be very important to some of the individuals that carry these genetic liabilities if a compound comes in at very low levels and disrupts the normal calcium signaling that goes on in a cell. So this is my chemical slide. So you're always making these choices, right? So if you want no PCBs, you pay a premium now, right? So you buy ocean fish. And if you're really unconcerned, you buy farm-raised salmon that's not organically fed, and that's $2.79 a pound. So. Okay. So what is it that I'm talking about dioxin-like? So dioxin-like compounds essentially, as their namesake would suggest, are very planar compounds. They're very persistent in the environment. And in fact, dioxin is the prototype. It's exquisitely toxic, acutely as well as chronically. It's well known that there may be an increased risk of cancer with very low level exposure, and as you saw with the Ukrainian president, can lead to some very painful consequences at higher doses. PCBs and dibenzofurans have been known for a long time to have some of the structures, of the many, many structures that were released into the environment that model dioxin and so are dioxin-like. They have similar activities to dioxin. In fact, they have the same target. Non-dioxin-likes, nobody cared about. That's because they had no dioxin-like activity. So they were generally considered to be of less concern. And so these include the polychlorinated biphenyls, which have substitution patterns of the chlorine, which makes them non-coplanar. So they don't sit like this in space, they sit like this. Makes them really bad ligands for the AH receptor, which is the dioxin-like receptor. But maybe they have other effects that we didn't recognize. This is of concern because even though we've banned these, it turns out, as I'll show you in a second, these are the major forms that we now find in the environment. the vast majority of PCBs that are outgassing from Richmond sediments just in the Bay Area are non-coplanar PCBs. These have been measured over Chicago because of outgassing from Great Lakes. And there's a huge concern in the Hudson River now because GE has been forced to dredge all of those bad PCBs they dumped in there over the many, many years. There's concern that they'll mobilize and release to the environment. But we have other non-coplanar compounds that have made it to market that we didn't have any reason to ban or prevent to get to market. One of the group is the brominated flame retardants, which we're all being exposed to right now. I think, maybe not. I think maybe they took great care not to put them in these foam seats or in the carpets. But in general, these are all over the place. You've all heard about them. And another one, which may be a surprise to you, which we've received some resistance from the company when we published the paper last year, is the antimicrobial triclosan. So triclosan is now in your toothpaste, in your socks, in pizza cutters, underarm deodorant, and of course antibacterial soap. As you see here from its structure, it's non-coplanar, so it has no dioxin-like activity. So the target we're looking at is present in neurons, and this is just all the little red spots are the target of these non-coplanar PCBs. They're called ryanodine receptors. They're very strategically located. As you can see, they're not all over the cell. They're in just very, very localized regions where they perform a certain function. And this is what the PCBs does to them. So just look over here. This is a channel that's a single ryanodine receptor. It's a calcium channel, which we've closed by decreasing the calcium in its environment. This is the way these channels should look, and occasionally they open when they're needed to release some calcium to do some function. This is that same channel on PCB. So you notice that here, the channel is closed most of the time. In these traces, they're closed all the time. If you put a relatively high concentration of PCB in the immediate environment of this channel, it sticks wide open. And so now there's unregulated calcium flux through this channel, and you can see here that it's open most of the time, whereas this one is closed most of the time. Now, this is of basic interest, but it shows that, in fact, there is a molecular interaction with non-coplanar PCBs with this very large protein, and you can actually see what it does at molecular detail. So this is the closed channel, which is this channel here, and you see that it's reorganized. Here's the pore right in the center where the calcium flows through it when the PCB is on board. So we know at the molecular level that these two interact. Does this have an implication? I'm going to go past the video here because it's getting late. Come on. Do we know that these compounds are in abundance in human samples or in sediments? Are we exposed to these compounds? So here is a biochemical assay that scores the 28 most active compounds, and we just matched them up because the data was available with Fox River fish, the PCBs in Richmond, California sediments, just down the road in Berkeley, and serum levels found in the Akwesasne Mohawks in upstate New York. And the take-home message here is that even though we've only tested about 25 compounds, they represent about 50% of the body burdens or the sediment burdens. So they are relevant to environmental exposure. What do they do? What do these compounds do outside of just interacting with a molecular target? So in the hippocampus, where we think autism manifests many problems, this is a region of the nervous system that's very important for memory. We can make measurements. Again, this is now in a rat model. So this is the hippocampus of a rat. We've put it on an electrode array, and we measure excitability. So if you chug along here from left to right in time, this just shows the excitability of the cells at rest, or when you put a vehicle, which is a control that this compound will be ultimately dissolved in, it doesn't affect the excitability of the neurons in the hippocampal slice. Now, if you expose these hippocampal slices to a GABA receptor blocker, That is, you block the inhibitory side. And I didn't show you this, but there's a lot of evidence suggesting that GABA receptors, inhibition in the autistic nervous system, is deficient. And there are many chemicals that hit that target, but I'm not going to talk about them here. But here, we just did the poor man's autism model where we block the GABA receptors, and the way we stimulate these guys, it shouldn't have an effect on the resting slice preparation. And it doesn't, because the control, you can go from left to right. It's pretty much baseline. There's a little bit of elevation, but it's about 15% at the most. Now if you come in and you perfused one of the most abundant PCBs on the slice, what you see is, in fact, you start to get excitation, and then there's runaway excitation. In fact, now you've unmasked the effect of the PCB, and if you look at the gray waveform here, this is what we call the seizure-genic waveform. So in other words, they're after potentials which are models of seizure disorder. So we think that this is significant. Why is it significant? These concentrations are unheard of. We used these concentrations within the last 2 or 3 years in the lab. Before that, we published a paper in 1996 where we were looking at similar compounds. We were looking at 10 micromolar. And we saw this flash, and then the slice just kind of shriveled up, and we said, it inhibits. But people said, well, that's just toxicity. It's not relevant to neurodevelopment or balance of excitatory inhibitory circuits. Here we're working at about 1,000 times lower concentrations. So you induce a deficiency in the GABA system, you now see very, very potent effects. And they're excitatory, of course. What does that mean to the developing nervous system? So you have to do a developmental study. And so we exposed rats, in fact, pregnant rats at gestational day 5. We just put a little bit of PCB, in this case a particular PCB that has absolutely no dioxin-like activity. We put them on a little wafer. They love it. They gobble it up. And you do this once a day to expose them over the life of the gestation and lactation period. You never expose the pups directly. What does this do? Well, Tal Kennett and Mike Merznick were very interested, and this is a collaboration that occurred many years ago. Tal Kennett's at Harvard now with Martha Herbert. What they do there is they map the auditory cortex. So this is the map of a rat auditory cortex, which shows high frequency to low frequency responses in the brain. So this is the integration point. This is where everything you hear gets integrated. And it's integrated according to the frequency. And as you can see, there's a very nice pattern in the auditory cortex. It's very organized. So you give the tones to the animal. At high frequency, this region lights up. You give them low frequency, this region lights up. And then you put it together and you make a map. What does it look like if you have PCB exposed animals? So here are 3 examples. You have some that seem to have the proper left to right organization, but you're missing a lot of fields. So some of the neurons or regions of the neurons don't make proper connections, or the neurons are missing. Here's one where the presentation is completely reversed. It's flipped. So here you have, instead of high frequencies, you have low frequencies, and here you have the high frequencies. But again, it's very disordered. As I talk to these folks that know something about this, what this might mean to humans, perception of sound and, of course, language is extremely important to the development of language. And so if you had this kind of problem in real, even at a lower level, it would be very, very, have impact. We've now done this. This paper just came out a few weeks ago in Environmental Health Perspectives, where we did treat the animals with, in this case, a mixture of PCBs, and now we asked them to learn something. And so here is a drawing of a typical neuron from a non-treated animal. This is the pups now, so they're very young. So this is the complexity before you train them over here, and after you train them, on average, there's a 22% increase in complexity. That's been established in the neuroscience literature. That's nothing new that we're showing. However, if you take the PCB-treated animals, exposed animals, their basal dendritic complexity is about 20% more on average. These are cortical neurons. These are mapped in situ while they're in position in the brain. There's about a 20% increase in complexity. But after learning, instead of increasing their complexity further or not changing, they actually crash and burn. They have a 17% decrease, so they've retracted. So these are not behaving properly. And these are unprecedentedly low exposure levels, and we pushed it. We actually went higher in concentrations, and now what you start to see, at least in normal rats, is that the effects are less. So we've turned toxicologists' prime credo of the dose makes the poison, and we said, well, maybe lower doses are more hazardous than higher doses. Why? Well, maybe at higher doses you induce protective mechanisms that you don't induce at lower doses, and so maybe lower doses could be more harmful. So that's something that a lot of people have suggested that we need to re-examine. So I'm going to stop here. Well, okay, one last scary thing for you guys. We can show that these effects can be, at least at the biochemical level, be replicated with certain flame retardants, especially the metabolites of flame retardants. In my lab, we're not so surprised because about 15 years ago, we were looking at a marine product that came out of a sponge out of Australia, and it turned out that the active principle in it was a very close to a brominated flame retardant. But this proves that some of the brominated flame retardants that we're concerned about are extremely active in our assay. They're not active in the dioxin assay. Triclosan. I've been quoted as saying triclosan causes autism. We have no proof that triclosan causes autism, and we have no proof that triclosan causes neurodevelopmental defects. I want to make that very clear. What we have missed is that triclosan is increasing in humans. This is not our work. This was published by the CDC about 2 years ago. And what you see here are human blood levels. These are human urine levels from 6 to 11-year-olds up to 60-year-olds. And as you can see, there is, in fact, a pattern of urine levels, exposure levels, and the variability is really large. What do you think the variability is assignable to? Well, one thing that it's assignable to is socioeconomic status. The cleaner you are, the more money you have, the higher the levels. At least this preliminary study would suggest that. Are these inert? Because other than a bacterial target, they're supposed to inhibit the bacterial enzyme, which is only found in bacteria, so they couldn't possibly be effective on a heart cell, right? In fact, any compound today that would be shown to have this activity in vitro would never make it to market. It would die in tox, is what they would call it. So what we see here is a cell that's been exposed to 500 nanomolar just during this time. And what you see where the red line is, and I don't have the control, I'm sorry, is it would be absolutely reproducible, no increase in the amplitude of the response. At 500 nanomolar, there's a very significant increase in amplitude. But as you push the concentration higher on different cells, these are not all the same cells, you see that now you get this transient increase, and then essentially the cell stops responding. These cells are not sick, these cells are not dying. These cells have lost their ability to couple meaningful signals from outside to inside the cell. So we think that this is significant. We think that if you talk about BCBs not being an issue anymore because the levels are declining, we're not making them anymore. We have to account exposure of certain populations that may be more susceptible, not only to PCBs, but to brominated flame retardants and to things like triclosan and other compounds which are non-coplanar and may have effects we've completely missed over the years. These compounds are more recent, so they've been heavily used only since about the early 80s. That's when a lot of our increase in prevalence of autism, our increase in prevalence of asthma, and our increase in prevalence of metabolic disorders. There are other things that contribute, of course, but we don't know that these don't contribute. And I'd be happy to take questions, or both of us, yeah.
want to know about autism in a homogeneous group, like the Somali immigrants in Minnesota, or Hutterites, or Pennsylvania Dutch. Because you keep saying it's heterogeneous group, but wouldn't we expect to be something very similar about a small group? Do you have any information about that? A couple of issues on that. You can drop it, the too complicated. Another one is the association of cerebral palsy with autism and the gastrointestinal system and its immunology in autism. Those things I'd like to learn some things about. That's right.
Do you want to take the immunology?
So maybe I'll start with the last one and work my way backwards. The GI system, there's a major role of the immune system in the GI system, as well as the nervous system. So that's you could say that the GI system is sort of like a point of convergence. It's highly innervated. There are a lot of, the nervous system is significantly present in the GI system, and the immune system, of course, is the GI system is the largest organ of the immune system. We're constantly eating things that could make us sick, and therefore it's a prime area for active immunology. So you could add to that then the stress, the role of stress and the effect on GI pathology. So it's definitely a major area of research, and I think it's not surprising that GI symptoms are common in autism, whether it be from immunologic phenomena, nervous phenomena, or a result of stressors that the child or the person with autism is experiencing. So with that said, there's a lot of room for more research in the connection between the GI system and autism. And certainly we see that in a large proportion of our cases that are enrolled in the studies that we have GI symptoms in autism. I am familiar with the Somali case. I've read up on that a little bit, and it's very important to be able to characterize the groups that we're using for research in autism. So we try to get as much information from the families as we can and try to, where possible, segregate based on similar backgrounds or whatever to any sort of information that could show a subgroup within autism. So it's very important to study groups that may be more homogeneous, but even doing so, although we don't have those data, even doing so with the data that we have, we still see a tremendous diversity in the symptoms, and so that's. And what was the second question?
No, it was about cerebral palsy.
Oh, the cerebral palsy.
Increased cerebral palsy with increase in palsy.
Yeah, I don't have any data on that. I've seen it, and but sorry, I can't comment on that.
Yeah.
I just saw, I came in late, I apologize, but is Dr. Daniel somebody, are you an MD or a PhD?
Neither. I'm almost finished with my PhD. I'm a researcher in Dr. Judy Vanderwater's lab.
Okay, because maybe my question won't, I'm not sure you can address it, but I've measured the C3A and C4A in the blood of autistic children, and a subset of them are elevated, and I wanted to find out if that is specific for Lyme disease or if other infections increase autoimmune disease, kind of fire up autoimmune disease. Because I thought those are markers, C3A and C4A autoimmune, and I know that they can be related to Lyme, but I didn't know about other infections.
Yeah, so the complement system, which those are two proteins involved in the complement system, which is a fundamental activation of the immune system. There have been a lot of findings. I don't know that there is consensus that there is the significant alterations across multiple studies in autism, although I have seen those reports.
It's just a subset. It's definitely not all.
Yeah, and that's one of the frustrating things is also during my presentation I talked about several things that we observe in smaller subsets of autism, and hopefully information like that will aid us in distinguishing between different groups within autism, especially for tailoring treatments. So.
I was wondering at the National Institute of Mental Health on pandas and, pediatric autoimmune psychiatric disorder associated with strep. And my son, we went through a number of tests and observations and determined this is what was going on with him. He developed tics and OCD and all these comorbid type of conditions. And I'm wondering if you guys have looked into anything like that.
Well, we have a lot of data on comorbidities, and the second study that I showed with regards to the maternal antibodies, which was the early markers of autism study, was done in collaboration with Kaiser and was this very large screen of families in Southern California. Ultimately, we were looking for significant associations for any sort of other comorbidities, and there were several, although not with pandas. And generally not with autoimmunity, either in the mothers or the children. There was some association, I believe, with gestational diabetes in the mothers, but it was not a highly significant association. So we certainly see these individuals, although when we try to do larger studies and look for associations in them, they tend to segregate out with smaller groups within the study and not the whole study.
I just learned something about a study that involved vinyl floors, and there was exposure from the vinyl floors. Do you know anything about that? Can you talk about that a little bit?
I think that just came out not too long ago, and I know virtually nothing about it. So sorry.
I was wondering if some of these chemicals can cause dysregulation in the way that people can take in certain things like calcium. Could that potentially explain why some people feel a casein-free diet, which has heavy levels of calcium, makes it?
It's a very good question. I don't think we have an answer to that. I think there are several known disorders. So if you go to something a little more cause and effect, so the one I think about, hyperkalemic periodic paralysis. It's not a calcium channel problem, but it's a sodium channel problem. So there's a mutation, and it's amazing. Individuals that have this mutation are more or less typical, unless they ingest a banana or something else that's really high in potassium, which tends to depolarize just a tiny bit. Most of us don't even feel it because there's really no clinical consequence. But those with this inability to inactivate these channels essentially undergo a periodic paralysis. They can't move, they actually fall, and then when they metabolize and their potassium levels go back to normal, they get up and they can. So, that's an example of an environmentally triggered syndrome. Whether this has anything to do with calcium levels, blood levels, I would tend to say not, because calcium levels are so highly regulated, but if you're super sensitive, just a tiny little change could, in fact. There are anecdotes of using some calcium chelators but they're very dangerous because you can't mess around with your calcium levels. So. Any other questions? Yeah.
Quick question. It's really specific, but this is from the first talk. It was kind of in last year pilot study with the primates. Were the controls, were they also injected with IgGs from human maternal as well, or were they just injected with just the used to inject
Yes. No, thank you. That's a very important question. And in this pilot study, we had so few animals to work with that we had a saline injected control to sort of control for the procedure, but we didn't have a non-autism IgG control in that part. That was the
control. There was maternal control and maternal from autism. The IgGs.
I'm sorry. Yeah. So there was the Yes. So, yes, to answer your question.
That's my role as a co-mentor on his dissertation.
We have a lot of studies going on, and I apologize for confusing there. But so, yes, there was a non-autism IgG control, and those were the control monkeys that you saw.
You were saying that the protein's not quite identical. Has there been, I know it's really difficult because I was working with something completely different, autism, not autism, Alzheimer's disease. But I know some proteins are, membranous are very difficult to isolate. But has there been any attempts yet to try to isolate these proteins that you found, the bands that you found yet? Or is that something in the works? Or that might be interesting if you could try to pull down those proteins that are identified or have something done.
And furthermore, they're probably not going to let me graduate until I figure out what it is. So that's, I am very actively working on that identification. I'm not sure why it's been so difficult, but it's, it has been challenging. But yeah, no, we are working diligently on that.
Any more questions? Thank you very much.
Thank you.
The UC Davis Mind Institute began in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, learning disabilities, and other brain disorders is helping children achieve their fullest potential. Please call or visit our website to find out more about current studies, our research team, and upcoming events.